mass cytometry is a powerful tool for being able to analyze complex biological samples and really resolve their single-cell heterogeneity. And this is, of course, a useful strategy when applied to large patient cohorts to be able to take, a single sample here, for example, just a whole blood sample, resolve a range of different immune cell populations, as well as their associated marker expression patterns, and then evaluate this across multiple different samples to be able to see either how cell populations vary in their frequency. So in this case, we're comparing longitudinal samples collected either during acute infection or at recovery. And you can see that plasmacytoid dendritic cells go down in frequency, dendritic cells go up. And we can also look at relative marker expression levels. So here, for example, we're seeing that the median intensity of CD40 is elevated on CD14 monocytes at the acute phase of infection. So these are sorts of the kinds of studies that many folks are engaging with using mass cytometry, and it's well suited to do these sorts of analyses. But an important caveat when considering this is that there are many sources of variability in cytometry experiments. So for example, there's the instrument can contribute levels of variability. There's variations in the antibodies that are used from day to day in staining protocols, or simply the volume of antibodies being pipetted on a given day. There can be sample matrix effects when dealing with whole blood. So for example, compounds in a given individual's plasma that may be impacting staining quality. And lastly, there's actually true biological variation between samples. So what we mostly care about is this last point here, the true biological variation. That's kind of what we're attempting to get at. And the challenge with these first four things is they all contribute to technical variation. And this technical variation can lead to type 1 and type 2 statistical errors, so false positives, false negatives, and ultimately these are the kinds of things we want to minimize so that we can really focus on that true biological variation.